Your reaction to this environment isn't unique. Most people, even non-scientists, are fairly blown away by the diversity and complexity. Hi, my name is Eric Mattson, and I'm a postdoc in the Department of Environmental and Science Engineering here at California Institute of Technology. My work under the direction of Professor Jared Ledbetter focuses on the microbial interactions and diversity of species that inhabit the termite hindgut. In particular, we're interested in some of the biochemical pathways that go on, and especially are interested in the process of acetogenesis, whereby these microbes consume hydrogen and CO2 gases and convert them to acetate, which is a key nutrient of the termite host's energy metabolism. Myself and others in the laboratory have developed targeted primer sets to be able to go in and assess key enzymes in this pathway, but in order to do so requires us to generate clean DNA samples of the community DNA in order to use for targets for this process. So today I wanted to demonstrate how we dissect the termite species that we're talking about, extract the DNA from the hindgut, and then purify it for use later for investigations. Today we're going to focus on one species of termite in particular called Zootermopsis nevadensis. This is a California dampwood termite collected locally here, and it's a rather large species, which makes it pretty easy to do the dissections and extractions from. Okay, so today I've already prepared some of the supplies that we'll need for the procedure. One of the first things we need is a certain amount of ice to chill down the insects that we'll be dissecting. I've also pre-chilled some TE buffer. This contains a compound that will help stabilize the DNA once it's released from the cells. In here I've got sterile vials preloaded with zircon